Welcome to AP Biology. Today I want to go over glycolysis, which is the first step of cellular respiration. And glycolysis is pretty much what it sounds like. So glyco, you might remember that, is something that has to do with carbohydrates. So the glyco part in this case means sugar or glucose. And lysis means to break. You've heard that with hydrolysis, which was the process where you use water to break something. Anyway, glycolysis or glycolysis means to break sugar or to split sugar. So sugar, you might remember, is generally what we're going to start with for cellular respiration. You can use other molecules for fuel as well, but it's easier to talk about glucose to start with, and so that's what we're going to do. Here's glucose's formula, C6H12O6. So that's 6 carbons, and so I've got a 6-carbon molecule right here. And if I'm going to do glycolysis, I'm going to end up breaking it in half like this. And so you started with 6 carbons. Each red bead is supposed to represent a carbon, and there are OHs and Hs sticking off these. But when you break it, you get these 2 molecules, each with 3 carbons. 1, 2, 3. And so that is what's going to happen. So here's the overall reaction. There are a bunch of little steps, but this is the overall reaction for glycolysis. You start with glucose, and remember each bend has a carbon. Oops, each bend is a carbon here. And carbon makes 4 bonds, and it's not showing you 4 bonds. That means there must be an hydrogen sticking off too. So each of these is a carbon, 1, 2, 3, 4, 5, 6. That's C6. And then the oxygens are shown. Here's one. There'd be another one here, 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 and here. So that's 1, 2, 3, 4, 5. That's 6. So that's the O6. And then the H's, I'm not going to draw them all, but you would have an H here, and here, and here, and here, right? Each oxygen has 2 bonds, etc. So here's glucose. And if you break that 6-carbon compound in half, you get 2 molecules with 3 carbons each, and they have hydrogens and oxygens still. So you've also taken off a couple of waters, and you make a couple of ATP. So this is an energy conversion. You're converting glucose into ATP. You've only made 2 ATP, but that's better than no ATP, right? This term, pyruvate, let's change colors here. You break down glucose into 2 molecules of pyruvate. That's this or this. Another word for pyruvate is pyruvic acid. I want you to know these vocab words. You're not going to need to know every single intermediate in the process, but because glycolysis starts with glucose and ends with pyruvate, that's really the final product of glycolysis. I do want you to know that term. And you'll hear pyruvic acid, same thing. Meanwhile, another thing that you've done is ripped off some hydrogens and some electrons too. And you might remember those electrons are energetic. And so what happens to those is going to be key. So we're going to talk about that. Oh my God, look at that. I just gave it away. I guess thought I was going to do a little bit later. Those electrons end up being carried by our good friend here, NADH. So we make a couple of those. So those transport molecules, those delivery molecules, are going to take electrons from glucose. It's going to take electrons and some hydrogen and transport them somewhere. And that is the mystery. Where is it going to go? Where should we bring those? I'll tell you someday. Okay, glycolysis occurs whether you have oxygen or not. It doesn't need oxygen, so it is anaerobic. So you need to know that anaerobic means no oxygen. And it happens in the cytoplasm. So I have this really old dude here because I want you to remember that glycolysis is ancient. It's present in every life form on the planet. So bacteria, archaea, all the different eukaryotes. Everybody does glycolysis. Not everybody does aerobic cellular respiration. There are bacteria that do fermentation. Yeast do fermentation. There are bacteria that only do anaerobic cellular respiration, but we're talking about the process of aerobic cellular respiration right now. Starts with glycolysis, and I'll just say everybody does glycolysis. After glycolysis, we'll continue on with something called the Krebs cycle and then the electron transport chain where we'll use oxygen, but some bacteria will continue on with a different process, but everybody starts here. So I want you to memorize that it happens in the cytoplasm, which I think makes sense because the cytoplasm, like everybody's got cytoplasm, right? Not everybody has mitochondria. So here's your mitochondria, and that's present in eukaryotes only, right? Plants, animals, fungi, protists, but not in bacteria. And so if I say everybody does glycolysis, maybe that will help you remember that it happens in the cytoplasm. Now, there are bacteria, plenty of bacteria that do aerobic cellular respiration without a mitochondrion, but they just do it on their cell membranes. So anyway, glycolysis happens in the cytoplasm, and I want you to have that memorized. It makes ATP with substrate-level phosphorylation. Phosphorylation means you put a phosphate on something. So you've got ADP, you add a phosphate, and you make ATP. Substrate level means it has something to do with an enzyme. So we'll get to that. Okay, evolutionary perspective, the early Earth was anaerobic. Before we had cyanobacteria making oxygen, the atmosphere was anaerobic. And so this process, because it occurred so early, it occurred without oxygen, so it's an anaerobic process. So life on Earth first evolved without free oxygen in the atmosphere. Energy had to be captured from organic molecules without oxygen. And so the prokaryotes that evolved glycolysis are the ancestors of all modern life. So everybody on the planet right now, all life forms do glycolysis. So all cells still utilize glycolysis, and that's important. All right, so here is the one other kind of weird thing. So the whole point of glycolysis and cellular respiration is to make ATP. But one wacky thing that happens is that you have to use some ATP. So you use 2 ATP in the process, you'll phosphorylate something else, and then you're going to make 4 ATP. And also that delivery molecule, remember, we'll draw that like a delivery guy here. He's bringing flowers around. No, he's bringing electrons around. Anyway, and then you end up with the pyruvate, which are those 3 carbon compounds. So, if you have a business, like maybe you mow lawns and maybe it costs $3 in gas, right? But then maybe you make, I don't know, like $10 overall. So that would mean the net would be $7. So you would be making $7, but you've got to invest in the beginning. So it's very similar here. We're going to call this the investment phase. You have to use 2 ATP in order to make 4. So overall, your net number of ATP is 2. Net 2 ATP. And then you also make these delivery molecules, which will be really important in aerobic cellular respiration. Okay, so now what we're going to do is look at the hairy details. Anyway, there's a bunch of intermediates. I want you to know that they're phosphorylated intermediates. I'm going to tell you the names of them and show you the structures. You don't need to memorize every single little name of every molecule. Okay, so here's glycolysis. The first step is where you use ATP. And so here's the ATP that you're going to use. And you break that down to make ADP. But the extra phosphates here are going to go on to glucose. So now glucose gets phosphorylated. And that step is going to require an enzyme, and you do not need to memorize the name of that enzyme. Yay. Okay, the next step is that you cut it in half. And so when you cut this in half, you have another molecule, pGal. You have another enzyme that does that. This is a 3-carbon molecule with a phosphate. So these pink things are supposed to represent the carbons. And of course, like if this is glucose that you start with up here, each of these circles is supposed to represent the carbons. There's also hydrogens and oxygens that I'm not showing you, but I'm just sort of doing this to make it maybe a little easier. Okay, the next step is that our delivery molecule, NAD+, is going to rip off some electrons and hydrogen from this molecule. And when it does that, it's going to stick a phosphate group on the other side. So now this is like a double phosphorylated molecule. So the energy from this split is used to combine inorganic phosphate with the molecule that you had before. The p-gals also give some electrons and energy to the delivery boys. So you start with NAD+, they rip off some electrons, you end up with NADH, right, and a hydrogen ion. So what are you going to use those NADHs for? We'll talk about that later. But right now what you've done is phosphorylated both sides of these. So I'm showing you this reaction and this reaction because for each one glucose, you're going to end up with 2 of these p-gals. Okay, so then the next thing that you do is you take the phosphates off of here and here with an enzyme, and you put them onto ADP to make ATP. And so that's called substrate-level phosphorylation. You can make 2 ATP out of this because see, you've made 4. 1, 2, 3, 4, but you had to start with 2 in the beginning, so 4 minus 2. Overall, you've made 2. Okay, so substrate-level phosphorylation. ATP is formed when an enzyme transfers a phosphate group from some substrate onto ADP. So that substrate is right here. This molecule is your substrate. You take an enzyme, and here's that molecule. You can see the 3 carbons in it. It's got a phosphate on it, and you're putting that phosphate onto ADP, and you'll make ATP. Yay! So substrate level phosphorylation is important because you only make a little bit of ATP from it, and I want you to be able to contrast that with oxidative phosphorylation when we get to the end of this unit. Okay, so to summarize glycolysis, what you do overall is make 2 ATP and 2 NADHs. So you put in glucose, you use 2 ATP, right? You need to have NADs to start with also to take the electrons. So these NADs produce NADH because the NADH rips electrons off glucose. You make 4 ATPs. So because you had to use 2 and you make 4, you only get a net of 2 ATPs at the end. You've broken glucose in half and you have this thing called pyruvic acid or